Hi, my name is Titouan Loriol, and in this video I will present to you the GeoLifeCLEF2021 challenge. The aim of this task is to learn a function which maps GPS coordinates to a list of species present at that given location. To do so, environmental data and remote sensing imagery at this point is given as input, and the model must infer from it the species living together at that location. Knowing where species are present is useful for a lot of applications, such as biodiversity conservation, land use planning, education, etc. But it's also closely related to traditional problems studied in ecology, such as joint species distribution modeling, species assemblage, measurement of diversity, and a lot of others. There are a few challenges inherent to this competition. First, due to the opportunistic nature of the observations, there's a high sampling bias. Secondly, we aim to predict sets from single labels, which is a non-standard classification task, and usually referred to as set-valued classification. Finally, we are using remote sensing imagery in complementary to traditional environmental variables. In the course of this year's challenge, we focused especially on this last point. I'll present briefly the data. There's different types of observation data used in the literature. Here we'll be focusing on presence-only data coming from citizen science projects, namely PlantNet and eNaturalist. We have nearly 2 million observations of over 31,000 plant and animal species. These observations are distributed across France and US, and the species distribution is highly imbalanced, as you can see on the figure on the right. So each observation consists of the observed species name and the location given as the GPS coordinates where this species have been observed. Traditionally, environmental models make use of bioclimatic and soil variables, which are, however, available only in a low resolution. Here we complement this type of data with high-resolution remote sensing images, such as RGB and near-infrared images, but also altitude and land cover data. To evaluate and compare the models, we need to make sure of a few things. First, we need to limit the effect of spatial autocorrelation, and so the splitting is not done randomly on the observations, but rather take care of the spatial distribution of these observations. Secondly, we use a set-value classification metric. However, as stated previously, this year we we keep it simple and use a top 30 error rate. Here are shown the results of this year's campaign. You can see in orange the baseline we provided, and in blue the runs provided by the participants. So as you can see, the best performing models is doing much better than the constant baseline on the right. The first outcome of this challenge is that CNN together with remote sensing patches outperform traditional models. This means that this type of data has important information which explains the species composition, and that moreover CNNs are able to capture this information. The second outcome is that it's necessary to aggregate properly covariates. As you can see, some models using all the covariates perform worse than some models using a single one or only a handful of them. Finally, here are some perspectives. First of all, the top 30 error rate is still high, around 75%. So there remains considerable room for improvement. In particular, one open question is how much complementary or redundant is remote sensing data compared to bioclimatic and soil data, and how to make use of all the different covariates we provide and aggregate them properly. Beyond this challenge, the dataset is available for research, and any contribution and feedback is welcome. Thank you for your attention. Here's a handful of useful links. In particular, there's the dataset paper, which presents into more details the dataset and how it was gathered, and the 2 overview papers, which presents into more length the results of the last year and this year competition.